Hello everyone. Today in this presentation I'm going to discuss about the topic vulva formation in C. elegans. But before going to the topic, I would like to mention that this presentation is a part of the initiative taken by Zoological Society of Assam. Now here I have mentioned learning outcomes, that is the content of the presentation. And after going through this PPT, the students will learn the following, that is about C. elegans and also about its anatomical structure, and they will also get to know why C. elegans is used as a model organism, then about the life cycle of the organism, and after all this they will finally get to know what is vulva and its mechanism of induction and different signaling pathways involving here, and also about some of the mutation cases that can affect vulva formation. Now coming to the first point, that is introduction of C. elegans. Here we will discuss few characters about the organism. C. elegans is a free-living microscopic nematode which is non-parasitic in nature. It is small in size, that is about 1 millimeter in length. It is also known as roundworms, which is transparent, and it was the first multicellular organism to have its whole genome sequenced. C. elegans mostly lives in temperate soils environment and mainly present in two sexual forms, that is a hermaphrodite and a male. Most of the C. elegans are hermaphrodite, that is they contain both male and female sex organs. So in the next slide we will discuss about its anatomical structure. Now coming to the anatomy of the organism, that is anatomical structure of C. elegans. So the basic anatomy of C. elegans include a mouth, pharynx, intestine, gonads and rectum, etc. Like all nematodes, they have neither a circulatory nor a respiratory system. As we know that there are two sexual forms are found on C. elegans, that is a male and a hermaphrodite. So in case of males, they have a single-lobed gonad, a vas deferens, and a tail specialized for mating, which incorporates spicules. As it is an hermaphrodite C. elegans, here it has ovaries, oviducts, and spermatheca, and a single uterus. And in this diagram, it is clearly visible the position of vulva development also. So with this particular diagram, we can clearly understand the anatomy of the nematode and all the position of the organ. Here we will discuss about why C. elegans is used as a model organism. C. elegans is studied as a model organism for a variety of reasons. Here I have mentioned few of those. So as we know that it is a multicellular organism that is simple enough to be studied in great details. Second, C. elegans is transparent, which facilitating the study of cellular differentiation and other developmental processes in the intake organism. In addition, the organism has short generation time or short life cycle, and it is very easy to cultivate. Again, C. elegans having its whole genome sequenced, and it is also easy to preserve and very cheap to maintain in laboratory. and it also has large progeny size. So due to all the mentioned reasons, C. elegans is studied as a model organism. So in the next slide, we will discuss about its life cycle. Now here we will learn about life cycle of the organism. So an adult hermaphrodite C. elegans produces roughly 300 eggs, and after hatching they pass through the 4 larval stages. As we can see in this particular diagram, the 4 larval stages includes L1, L2, L3 and L4. And L4 larvae finally molds into adults. Again, this L1 larvae may proceed through the alternate Dewar pathway under harsh environmental condition. As you can see in this diagram, the L1 larvae may undergo the alternate pathway. This alternate pathway, but this only happens under harsh environmental condition, that is in crowded environment or in the absence of food. This Dewar larvae are adapted for long-term survival and dispersal to new environments. So once a more favorable environment, this larvae re-enter reproductive development by molting into L4 larval stage and progressing through the rest of the life cycle normally. At 20 degrees Celsius, the C. elegans has an average lifespan of approximately 2 to 3 weeks and a generation time of approximately 4 days. So here we discussed about the life cycle of the particular organism, that is C. elegans. So from the next slide, we will study about vulva and its mechanism of induction. So now what is vulva? Vulva is a part of the female sex organ in the hermaphrodite roundworm that consists of 22 cells and which serve as a structure through which sperm enter and fertilized egg leave the gonad. And if the vulva develops in the wrong position, then the gonad will not receive sperm and the egg cells never fertilize. So correct position for vulva development is very important. So the positional information needed by the vulva progenitor cells is provided by a cell within the gonad called anchor cell, which gives the inductive signal here. So remember that the vulva is a part of the female sex organ and which is formed by 22 cells. So we have to study about the formation of these 22 cells, which will later form vulva. Now in this particular slide or in this diagram, we will discuss about the mechanism of vulva induction, that is the formation of those 22 cells in C. elegans. Vulva development in C. elegans involves a network of intercellular signaling, signal transduction and transcriptional regulation. Let's start with the first step of it. So the process begins with the formation of 6 hypodermal precursor cells or vulval precursor cells, also known as VPCs, which are numbered as P3P to P8P, that is P3P, P4P, P5P, P6P, P7P and P8P, total 6 vulval precursor cells. And these 6 cells will take part in vulva formation. And each of these 6 cells have the equal potential to contribute in vulva development by receiving the needed signals from another group of cells. Means these 6 precursor cells receive signals from other cells for the formation of vulva cells. And the positional information needed by these 6 VPCs to get divided into the respective parts of the vulva is provided by the cells called anchor cell, this anchor cell, which act as an inducer here. So this anchor cell will act as an inducer for these 6 VPCs. So the first signal the VPCs received is given by this anchor cell. The anchor cell, which is present just beneath the gonad, will secrete a protein called lin-3 protein. So this anchor cells will secrete a protein known as lin-3 protein. And this lin-3 protein, which will bind with its receptor present on VPCs, that is LAT23 receptor. So the receptor for this lin-3 protein is present on this VPCs. And those VPCs are nearest to this anchor cell will receive high amount of lin-3 as compared to far ones. So here P6P, this P6P is present near the anchor cell. So it will get higher lin-3 protein, and remember that the cell who is getting high lin-3 protein from the anchor cell will develop the primary cell or central cells. So P6P will form the primary or central cell and later divides to form into 8 new cells. Now P5P and P7P, these 2 cells are present adjacent to P6P, will also get a moderate concentration of lin-3 and develops into secondary or lateral cells, which will later give rise to 7 cells each. So finally 8 from these primary cells and 7 from each lateral cells will finally form 22 vulva cells. Now other remaining 3 VPCs, that is P3P, P4P and P8P, will form hypodermis because they are not getting lin-3 protein from the anchor cell, because we know that lin-3 is required for vulva formation. Now look at this diagram. Here mention another protein, that is lin-12. In addition to forming the central vulva cells, this P6P cell also signals laterally to the 2 adjacent cells, that is P5P and P7P, and instruct them to generate secondary or lateral vulva cell. The P5P and P7P cells receive these signals through the lin-12 protein on their cell surface. Stimulation of lin-12 on the P5P and P7P cells induce expression of a phosphatase that inactivates MAP kinase pathway, as because MAP kinase pathway is for primary VPC formation. So, as we have discussed in the previous slide about the mechanism of vulva induction in C. elegans, in this particular slide, we will discuss about the signaling pathways that occurs in vulva formation. So, signaling pathways are mainly of 2 types, that is inductive signals and lateral signal. So, here we will discuss about inductive signal. We know that the anchor cells are very important as it acts as an inducer by giving inductive signals to VPCs in the form of protein called Lin3 protein, which is similar to epidermal growth factor found in human body or other higher animals. In the absence of the anchor cell, a vulva doesn't develop. So these are extremely important. Now the Lin3 protein that is secreted from the anchor cell binds to the receptor known as LAD23 or EGFR, that is epidermal growth factor receptor, present on the VPCs, and the signal is transferred to the nucleus through the MAP kinase pathway. Now the highest amount of Lin3, that is the highest amount of Lin3 is received by P6P. This P6P will receive the highest amount of Lin3 protein secreted from these anchor cells and adopt primary fate and forms central vulva cell. This inductive signal includes RUS or MAP kinase pathway, which is a cell signaling pathway. So in the next slide we will discuss about lateral signal. So as we discussed in the previous slide, that is those VPCs or vulval precursor cells which are present near the anchor cells, which will get the highest amount of Lin3 protein as compared to far ones. So this P6P being closest to the anchor cell receives the highest concentration of Lin3 protein and generates the central vulva cell or primary cell, and which divided to produce 8 new cells. Whether the other two VPCs, that is P5P and P7P, receives a lower amount of Lin3 protein and hence develops lateral vulva cells. So this P5P and P7P will form the lateral vulva cell and develops into 7 cells in each. So here we are discussing about the lateral signal that is given by P6P to P5P and P7P and instruct P5P and P7P to generate lateral vulva cells or secondary cells. So this is how 22 vulva cell forms. This lateral signal involves Lin12 and Notch signaling pathway. As we already discussed that the remaining VPCs, that is P3P, P4P and P8P, adopts tertiary fate and develops hypodermis, that is the external skin layer of the roundworm, because these three cells are present too far from the anchor cells. Hence they get very small amount of Lin3 protein from the anchor cells. Now we have discussed the signaling pathways, that is the inductive signals and the lateral signals, and how they involved in the vulva formation. So in the next slide we will discuss all this signaling pathway in a flowchart. Here this is the flowchart that is going to explain different signaling pathways that occurs on VPCs during vulva formation. So at first, see, there is a Lin3 protein which binds with its receptor, that is LAD23 receptor, present on the VPCs. Now those cells having LAD23 receptor on it will get a high concentration of Lin3 protein, because Lin-3 protein act as a ligand for lead-23 receptor. So VPCs having lead-23 receptor on it will get a high concentration of Lin-3 protein and form primary VPC or central cells. And next, see, there is inhibition of Lin-12 in primary VPC, and this is the main reason due to which primary VPCs are not converted into secondary VPCs. Now in the next step, the primary VPCs will activate DSL protein, and now this DSL protein is going to activate the Lin-12 inside secondary VPCs. Now Lin-12 is activated by this DSL, act on P5P and P7P and develops them into secondary VPCs. And another function of secondary VPC is that they used to inhibit lead-23 on it. As we know that lead-23 act as a receptor for Lin-3 protein. As because secondary VPCs inhibit lead-23 receptor on it, so they are not able to receive a high amount of Lin-3 protein and hence converting themselves into lateral vulva cell. Again, see, here is due to lateral inhibition by the primary VPC with Lin-12, there is a development of secondary VPCs. So Lin-12 is an important factor in successful lateral inhibition. So these are all the signaling pathways occurs during vulva formation, and in the next slide we will discuss about some of the mutation cases, that is how mutation can affect vulva formation. Now here we will discuss some of the mutation cases that occurs during vulva formation. So let's start with the first case, that is if there is low Lin-3, then how it will affect vulva formation. We know that Lin-3 protein is very important as it acts as an inductive signal on the VPCs to adopt primary VPCs. So in low level of it, there will be no primary VPC formation will occur. Now coming to the second case, that is if Lin-3 blocked or mutated. In this case also, there will be no vulva formation as same as the first case, because Lin-3 is important for vulva formation. Now third case, that is if anchor cells destroyed. See, in this case all 6 VPCs will form hypodermis, because as we have studied that anchor cells act as an inducer which secretes Lin-3 protein, due to which primary and secondary VPCs develops. So in absence of the anchor cells, all the VPCs will develop hypodermis and there will be no vulva formation. Now fourth case, if lead-23 blocked. We know that lead-23 act as a receptor for Lin-3 protein on the VPCs. So in case of lead-23 blocked, there will be no one to receive Lin-3 protein. So vulva formation. So in this case also, there will be no vulva formation. Now fifth case, that is if tertiary VPC or tertiary cells are destroyed. So we know that in vulva formation only primary and secondary VPCs are involved. The tertiary VPCs are responsible for hypodermis formation. So here in this case, vulva formation will take place. Now sixth case, that is if the gonad cells destroyed. Here see, there is no relation of gonads with VPCs and anchor cells because anchor cells are present beneath the gonad cells. So in this case, vulva formation will be normal. Next, that is the last case, that is if 3 central VPCs are destroyed. That is if primary and 2 secondary VPCs are destroyed. In this case, the rest of 3 cells will form vulva that was previously forming the skin or hypodermis, because now the gradient of lin 3 will be going to distribute on the rest of the VPCs. As we discussed earlier, all these 6 VPCs have equal potential for vulva formation. So here, if 3 central VPCs are destroyed, then the rest of the 3 cells now able to get the lin 3 protein, and this rest of the 3 cells able to form vulva. So at last, here I have mentioned some of the suggested reading and references that you can use for your further study regarding this particular topic. So if you have any doubt or query regarding the topic, you can contact me in the email address given here. Thank you for your kind attention.